A groundbreaking technique developed by NIHR-supported researchers could transform liver transplants by repairing damaged donor livers using lab-grown mini bile ducts. This cutting-edge approach may one day help patients with failing livers avoid transplants altogether. Bile ducts are the liver's built-in waste disposal system responsible for transporting bile, which aids digestion and removes toxins. Unfortunately, malfunctioning bile ducts are the leading cause of a third of adult liver transplants and 70% of pediatric liver transplants. Even after transplantation, bile duct disease can return in up to one-third of patients, leaving few treatment options. A major challenge in liver transplants is the severe shortage of suitable donor organs. Many livers become unusable due to damage sustained during cold storage. Seeking alternatives, NIHR Cambridge Biomedical Research Center scientists explored the potential of lab-grown bile duct cells to repair damaged livers. The researchers focused on creating bile duct organoids, also called mini-organs, using cholangiocytes, the cells that line bile ducts. For the first time ever, they successfully transplanted these lab-grown cells into damaged human livers, proving the concept that cellular therapy could repair and restore liver function. Their breakthrough involved using single-cell RNA sequencing to study individual cells lining the liver's bile ducts. Remarkably, they discovered that gallbladder cells, typically unaffected by bile duct diseases, could be reprogrammed into functional bile duct cells. The team cultured these reprogrammed cholangiocytes in the lab using organoid technology, enabling them to mimic the bile duct's natural function. They then injected these organoids into damaged donor livers using a specialized perfusion system. The results were groundbreaking. The transplanted organoids successfully integrated into the damaged livers, repairing bile ducts and restoring liver function. This marks the first time human donor livers have been repaired using lab-grown cells, offering new hope for addressing the donor organ shortage. Professor Ludovic Valliere, a senior researcher in transplant and regenerative medicine at NIHR Cambridge BRC, stated, "This is the first time that we've been able to show that a human liver can be enhanced or repaired using cells grown in the lab. We have further work to do to test the safety and viability of this approach, but hope we will be able to transfer this into the clinic in the coming years." This pioneering technique could significantly increase the number of viable donor livers, ultimately saving countless lives. Beyond liver repair, it lays the foundation for developing cell-based therapies for other organs and diseases, opening new frontiers in regenerative medicine. Subscribe for more videos like this. See you in next video.